Good morning, everyone, and thank you for being here today. I'm pleased to welcome you to this presentation on DNA barcoding and herbal drug identification. My name is Dr. S.K. Singh, and I hold a PhD in this exciting field. Today, we will explore how DNA barcoding is revolutionizing the way we identify herbal drugs, ensuring quality and authenticity. Over the next few minutes, I will walk you through the key concepts, techniques, and applications of this innovative approach. So, let's get started and discover the future of herbal medicine verification together. Today, I want to talk about DNA barcoding, which is an advanced molecular technique used for the accurate identification of plant species. It works by analyzing short, standardized regions of genetic material, which act like unique genetic fingerprints for each species. In the field of herbal medicine, this is particularly important because raw materials often go through extensive processing, and they may not have clear or distinguishable physical features anymore. Traditional methods, such as visual identification, can be unreliable in these cases. DNA barcoding overcomes these challenges by providing precise, species-level authentication through DNA sequencing. This means we can accurately identify the plant species even when physical characteristics are lost or ambiguous. This approach plays a crucial role in detecting adulteration, when incorrect or cheaper ingredients are mixed in, and in ensuring the authenticity of herbal drugs. By doing so, DNA barcoding helps strengthen our quality control systems, which are vital for safe and effective herbal products. As the demand for safe, standardized herbal medicines continues to grow, DNA barcoding is emerging as a powerful and reliable tool in modern pharmaceutical quality assurance. Let's take a closer look at the scientific basis behind DNA barcoding. Essentially, it relies on DNA sequencing to identify species. The process involves comparing the DNA from an unknown sample to reference databases that contain known sequences. This comparison helps us pinpoint exactly which species we're dealing with. Key regions of the DNA that are most useful for this purpose include the RBCL gene and the MATK gene. These regions are particularly informative because they tend to vary enough between species to allow for accurate identification, but are also conserved enough within a species. That's what makes them ideal for this kind of analysis. By analyzing these specific gene regions, we can achieve species-level identification, which is a pretty powerful capability. Now, what exactly is DNA barcoding? It's a molecular technique that uses short, standardized DNA sequences, kind of like a genetic fingerprint, to identify different species. Think of it as a unique barcode for each species, which makes identification quick and reliable. This method is especially popular for plants because it targets regions of the plant genome that are well suited for differentiation. Overall, DNA barcoding combines genetic data with database comparisons to streamline and improve the accuracy of species identification, fitting perfectly into our broader goal of understanding and conserving biodiversity. Let's take a closer look at the limitations of traditional methods used in herbal identification. First, there's organoleptic evaluation, which relies heavily on human perception, so it's quite subjective. Different people might interpret the same sample differently, leading to inconsistent results. Then, microscopy, although useful, has its limitations because it can't always distinguish between closely related species, especially once the plant material is processed. Another important point is phytochemical variability. The chemical composition of herbs can vary depending on growing conditions, harvest time, and processing methods, making it hard to rely solely on chemical profiles for accurate identification. Additionally, traditional methods often struggle to differentiate between species that look very similar morphologically, which is a big challenge in herbal medicine. This brings us to the need for more precise identification techniques. Adulteration and substitution are common issues. Herbal products are sometimes intentionally or unintentionally replaced with other species, which can be harmful or reduce efficacy. Many species also share similar morphological features, especially after processing, when diagnostic characteristics are lost. All these factors highlight the necessity for molecular methods that can provide accurate, reliable identification, methods that go beyond what traditional approaches can offer. This is crucial to ensuring the safety, authenticity, and efficacy of herbal drugs in the broader context of quality control. Now, let's take a closer look at some of the key advantages of DNA barcoding. First, it offers high accuracy and specificity, which means we can reliably identify species even when the samples are very similar. This is especially useful in complex biological or processed materials. Speaking of processed materials, one of its great strengths is that it works effectively even when the sample isn't in its original form, such as in cooked or dried products. This makes it a powerful tool for detecting adulteration and contamination, helping to ensure product integrity and safety. Another important point is that DNA barcoding is independent of environmental factors, so it's not affected by climate, soil, or how the sample was stored, making it a very versatile and reliable method overall. Now, shifting to the workflow itself, it starts with sample collection, gathering a small piece of the specimen. Then, we move on to DNA extraction, where we isolate the genetic material. Next, PCR amplification takes that DNA and makes many copies of the specific barcode region, so it's easier to analyze. After amplification, we perform DNA sequencing to determine the exact genetic code. This sequence is then compared against a comprehensive database to find a matching species. And finally, based on this comparison, we can accurately identify the species in question. This workflow illustrates how DNA barcoding provides a streamlined, precise approach to species identification, connecting directly to the overall theme of improving accuracy and efficiency in biological analysis. Let's take a look at some of the limitations we need to consider with these testing methods. First, they require specialized equipment, which can be a significant investment and might not be readily available in all laboratories. Additionally, the costs tend to be higher compared to traditional identification methods, so budget considerations are important. Another challenge is DNA degradation, especially in highly processed samples like powders or extracts, which can make detection more difficult. We also need validated reference databases to accurately identify species, and these databases must be comprehensive and reliable to ensure correct results. Moving on to turmeric, these methods are crucial for authenticating turmeric and detecting adulteration with other curcuma species. This helps us make sure that the product has the right therapeutic properties and isn't compromised by adulterants. For example, with ashwagandha, similar roots can look quite alike morphologically, which makes visual identification tricky. But DNA barcoding offers a precise way to confirm the genuine species, preventing substitution with inferior or counterfeit plants. In essence, while there are some limitations, these molecular techniques provide powerful tools to ensure product authenticity and safety, reinforcing the overall goal of quality assurance in herbal and botanical products. Let's take a closer look at the regulatory perspective on this topic. We're seeing increasing acceptance in the field of quality control, which is really encouraging. This shift is supported by major organizations like the World Health Organization and the U.S. Food and Drug Administration, which adds a lot of credibility and confidence to these developments. Additionally, there's ongoing work to integrate these standards with pharmacopoeial standards, which are currently under development. This integration is crucial because it helps create a more unified and consistent approach to quality assurance across the industry. Overall, these regulatory advancements are helping to ensure that the methods and standards we use are not only scientifically sound, but also widely accepted and trusted. This progress aligns perfectly with our broader goal of improving quality and safety in healthcare and pharmaceutical production. Looking ahead, there are some exciting developments on the horizon for DNA barcoding and quality control. One key trend is the ongoing development of comprehensive DNA barcode libraries, which will make identifying herbal ingredients faster and more accurate than ever before. Another significant advancement is portable sequencing technologies. These tools are becoming more accessible and user-friendly, allowing for on-the-spot testing rather than relying solely on centralized labs. This means quicker decision-making and more efficient quality checks. Integration with artificial intelligence and bioinformatics is also set to revolutionize the field. By combining DNA data with sophisticated algorithms, we can improve identification accuracy, detect adulteration more reliably, and streamline workflows. All these innovations aim to make DNA-based methods a routine part of herbal industry quality control. They will complement traditional techniques like chromatography and spectroscopy, providing an extra layer of assurance. The role of DNA barcoding in modern QC is not to replace existing methods, but to enhance them. It supports GMP compliance by ensuring product authenticity and safety. Ultimately, these trends will help us deliver higher quality herbal products, building more confidence among consumers and regulators alike. Let's wrap up with a quick summary of DNA barcoding. This technique is a powerful tool for identifying plant species by analyzing specific regions of their DNA. In the context of herbal medicine, it plays a crucial role in confirming that the plant materials are authentic and not adulterated or substituted with other species. The process involves extracting DNA from the sample, amplifying particular barcode regions, such as RBCL, MATK, and ITS, then sequencing these regions and comparing them to reference databases to verify the species. The importance of this method cannot be overstated. It helps ensure we're using the correct plant, which is vital for both efficacy and safety. It also helps detect fake or substituted herbs, protecting consumers and maintaining product integrity. Overall, DNA barcoding significantly improves the quality and safety of herbal drugs. However, it's important to recognize its limitations. For example, if the DNA in processed herbal products is highly degraded, like in powders or extracts, it might be less effective. That's something to keep in mind when applying this technique in real-world situations. So, to wrap things up, DNA barcoding is really a reliable and advanced technique for accurately identifying medicinal plants. It plays a crucial role in preventing adulteration, which is a major concern in herbal medicine, and it helps ensure that the herbal drugs we use are authentic and of high quality. This, in turn, boosts overall quality control in the industry. Of course, there are some limitations, like issues with DNA degradation or the need for comprehensive reference databases, but despite these challenges, DNA barcoding remains a powerful tool. Its ability to enhance the safety and effectiveness of herbal medicines cannot be overstated. In conclusion, DNA barcoding is a truly powerful tool for herbal authentication. It directly addresses major quality control challenges, such as adulteration, and it represents the future of scientifically validated herbal quality control. And when we integrate it with conventional methods, we create a more robust and reliable quality assurance system that benefits both producers and consumers alike. For further support, please don't hesitate to reach out to us. We're here to help with any questions or additional information you might need. Thank you so much for your time and attention today. We truly appreciate your engagement and look forward to assisting you further.